Hello. Yes, I hope you're excited for today. Right, here we are. Let's give a round of applause from the stadium. Yeah. So this is Nicholas A. Casado. I'm your host of the Biomedical Engineering News. And today is March 12th, 2021. And first I want to say welcome. And here is often I feel we forget where our food comes from. And today I hope to shed some light into, what departments of the state controls what goes into your bodies and how genetically engineered products are developed. Nanotech for agriculture is so benign that nobody even knows what comes from Crispr or Aqua Advantage Salmon. Between this and peptides, I want to know how biohacking works. It doesn't always require putting something in your body special, but specifically knowing how your body functions. So how nature always nurtures our development in science, biotech, blockchain, and silicone. So stay tuned. We've got some fun news that we hope to dive into right now. So I'm grateful that you're here. I want us to feel good about science and biotech. And we're going to have a great day. All right, first up, we've got nanotechnology to advance Crispr and this genetic engineering of plants. So Crispr, which we've known about for a while, I could probably pull up some news about it.
CRISPR-Cas is a powerful new technology that is revolutionizing biology. It's like cut and paste for DNA. Using CRISPR-Cas, or CRISPR for short, scientists can delete, tweak, or completely replace the genes of any organism. And they can do it more cheaply, easily, and efficiently than ever before. Researchers are already trying to use CRISPR to introduce genes for disease resistance into wheat, insert malaria-blocking genes into mosquitoes, and remove HIV genes from infected cells in humans. Scientists didn't design CRISPR themselves. Instead, they borrowed it from microbes, like bacteria and archaea. These microorganisms use the CRISPR system as a defense mechanism against invading parasites like viruses. It works like this. When a virus attacks a bacterium, it introduces its own DNA into the cell. DNA contains the instructions for life. It's made up of 4 different chemical units, which the cell reads like a code. Some viruses use their DNA to hijack the bacterium's cellular machinery and make more copies of themselves. They eventually burst out of the cell and spread to other bacteria. But with CRISPR-Cas, the bacterium can fight back. The Cas part of CRISPR-Cas is an enzyme that works like molecular scissors. The bacterium uses Cas to cut the invading DNA into 2, disabling the virus. Next, the bacterium inserts a section of the intruded viral DNA into a special area of its own genome. Over time, the bacterium uses this area to build up a library of bad guys. It forms a kind of parasite hit list, so that it can recognize them if they attack again in the future. The bacterium copies these sequences into related molecules called RNA. Each RNA guide is combined with a Cas enzyme, turning the molecular scissors into precise targeted weapons. Now, when they encounter a piece of DNA inside the cell that matches the sequence on the guide, Cas will snip the intruder DNA.
Yeah. If you're not watching live, this is all pretty generalized stuff. I think you can learn, besides through claymation, that CRISPR has some technology that that we're just curious how it works. I think I spoke to a recent colleague that uses CRISPR research in the lab, and he kind of just explains it's like super simple. And in between that and mRNA, I think we've got a lot on our plates. So we're editing the human body mostly through what we eat. I just watched a video on somebody pouring some Pepsi on a slice of pork chops, and all these parasites were popping out. And, you know, I was a bit confused by that. I was like, maybe it's fat or maybe... And it got me thinking, there is so many... there are so many parasites that we don't know about. Probably crazier than Toxoplasmosis gondii, yeah, I think we got a lot to learn. That's all I'm saying. And I'm just grateful to be doing this with you. And I hope we can learn a lot more as we go. So hang tight. We're going to dive into this a little bit deeper as we go forward. And so CRISPR on genetic engineering of plants. And how is nanotechnology of nanomaterials address these critical challenges? Well, I definitely wish this JPEG was a little bit better, but I think there is carbon nanotube based delivery. So that's number one. So DNA delivery could be, carbon nanotubes. We've talked about this with the blacker than black color. And, well, as this carbon nanotube that absorbs light, it can also be used as a DNA delivery and strong protein expression without transgene integration. So, this expression in the plant leaves can actually be a strategy to interfere with the RNA. So then there's DNA nanostructure based delivery, where they study the synthesis and systematic assessment of DNA origami nanostructures for their ability to deliver siRNA molecules to mature plant leaves. So in this graphic, if you're watching on YouTube live, kind of has a nice little graphic of a tetrahedron and nanostring, and this all goes into the mRNA cleavage degradation, and then you inhibit. So that's just two ways. This article actually goes into a bunch of ways. So it sort of talks about the particle bombardment of the protoplast transfer action. And then over time, it speaks about, more specifically, the delivery methods. Now, if this wasn't behind a paywall, I would pull it right up. So I can sort of read it. So carbon nanotubes are actually pretty new. And then there's the proton blast DNA nanotubes. Let's move forward. How about that?
Nope.
Yeah. All right, so these actual agricultural so CRISPR can actually do a couple things where it can affect the metabolic engineering, the biofuel, the agricultural, make it disease resistant, therapeutic biosynthesis, and then you can cover the crops in this biomass to actually make it more resilient. Oh yeah, this is gonna be good. Alright, so let's move forward actually. Because I feel like what I wanted to talk about was the genetically modified plants and animals getting more and more into our food supply. And what does that mean for us? Whether it is, just like I was saying before, whether it's including metabolic engineering, so you could actually change the molecular structure to have more vitamins. I'm not sure, make it more red. Alright, so these pigs are actually, along with other regulated livestock, are actually being reconsidered for the regulation of genetically engineering. Now, the Trump administration suggested that USDA should be covering genetically engineering animals. Well, the FDA has been the clearance for this. Now, looking at the difference between the USDA and FDA, there's a lot of common ground. So, I think a prime example of a genetically engineered animal is salmon, which in 2015 it was approved by the FDA for consumption, for human consumption. Well, a pig last December, it was approved for medical and food use. So, not sure what that means. Maybe Betsy Fries is the actual pig that was. So actually, Betsy Fries is a agricultural journalist, and she is also a pig. Maybe the pig's named after her. Okay, so what the issue is that this Aqua Advantage salmon, which I'm going to show you a picture of right now, it's twice the size of a regular salmon. It's pretty big, at least 3 or 4 times. Now, they call this a Frankenfish. This is developed by Aqua Bounty Technologies, which contains a gene that allows it to grow much quicker. Now, you can raise salmon in farms, and here's the kicker. The FDA actually has clearance on all fish, shellfish, and all seafood, except for farmed catfish. So, now the question is, it's being covered, I guess, by the FDA. I mean, what's the distinction between farmed catfish and other fish, farmed salmon? So the USDA actually has been urged to be removed from all this because it's not efficient for taxpayers. And yeah, maybe this makes sense. Remove the USDA from the FDA's clearance on fish, or maybe vice versa. Just, make it simpler. Because now you've got this Aqua Advantage salmon legal, and it's actually been a big debate in Northern California. Northern California has a bunch of salmon that they've tried to protect, and they actually forced the FDA to reconsider the approval of the Aqua Advantage, saying that the FDA had failed to show its own procedures by ignoring some environmental consequences of the fish. Now, when I was up in Stinson doing a little hike through the redwoods, I saw a bunch of post-its, little flags in the ground saying, oh, this is where salmon eggs are, and they're very protective of them. So, to imagine the consequence of introducing a frankenfish, I'm not saying that they're going to be farmed in the same way. How do you farm salmon? And I guess it's just in a giant... Alright, so there's a salmon farm, and I guess it's you just farm it in the ocean. Farming up the fish in the ocean. So you can find them both farmed and... So I guess they're probably along the coast, and they probably contained it. But what happens if this frankenfish gets out and actually destroys some of the salmon up in the coast of California? Now, commercial season is just kicking off. It's time for getting those fish. And I just got my own what did I get? I got these my fishing pass, my fishing license, so I can go fishing. And I feel like there's not enough regulation, but whatever. How does this affect genetically engineered humans? Well, if you feed them genetically engineered food, maybe then you are a genetically engineered human. Alright, so let's just move on. I'm good on this. Thank you guys. You're so patient. I appreciate it. Let's hear what we got next.
Medical emergency!
Alright, we've got a computational guide to lead cells down desired differential paths. So, alright, so you got cells. And this is very thick. I didn't read enough. Let's actually jump ahead to the next one. It's too chompy. Alright, we've got Octopus. And they're actually making smarter prosthetics. So let's bring this up big.
There are 230,000 marine creatures.
So just real quick, they're showing a bunch of cute little fish on the screen. And I gotta say, this is great news. Big fan of fish. They're so cute.
Don't know a lot about them. So my lab has been interested in understanding how nervous system of octopus works. Octopus is amazing in many ways. It has 3 hearts, it has blue blood, and it also regenerates many parts of its body. So there's a lot of interest in octopus research about how it regenerates.
Inside Science.
Ooh, this is a
People
fun
are
article.
developing robotic prosthetic arms that can move in many ways, quite similar to what a human hand can do. But still, we don't have ways to have prosthetic arms that are adaptive, that are flexible, that can change the direction midway. An octopus can do that. So octopus has 500 million neurons. The majority of neurons are in the arms, and each arm is a completely independent unit. Each arm has something like a brain in it. The octopus makes most of its decision in the arm. It tastes in the arm, it thinks in the arm. What we would consider things that we would do in our brain, octopus a lot of time does it in the arms. The octopus is really as an amazing model to learn a lot.
Now, I think that's super interesting. You've got octopuses thinking with their arms. Now, I guess that would make us reconsider what the human brain is, you know? So does neurons equate brain function? Soft tissue?
How it calculates the algorithms that it uses in the arm and how we could potentially translate into developing a new type of robots, a new type of prosthetics that will move flexible and be adaptive like an octopus arm. We record our octopus, each octopus in the tank 24/7, so we gather a lot of data about octopus behavior, the way it moves. So together, all the data we're going to gather, all the way from a single cell to electrophysiology, to motion capture, to the entire behavior of the octopus. We're hoping to identify the algorithms and the principles that can predict and explain how the octopus arm moves. And then we will take these algorithms and we'll feed it into a new type of a soft robotic octopus arm, which we hope will move and be adaptable. There is a lot to learn from marine creatures and understanding how different organisms develop completely different type of strategies to resolve different challenges that they have. It's just bound to fuel a whole new field of neuroscience and impact medicine.
Wow. All right. I hope you felt that, because I was feeling like some of the simulations they were doing in there was pretty interesting, you know? So like how an octopus arm can be simulated could possibly be used as a mechanism for how it thinks. So stretching the arm out in a certain way. I mean, I don't know how they communicate. I mean, it's three hearts, a beak, blue blood. It definitely opens up a lot of question of what does it mean to be what does it mean to be alive? You know, intelligent, right? Communication or All right, so let's get on with it. All right, we've got 3D printing, which actually could do a life-size hand in 19 minutes. Now, if you're listening to the audio, we're pulling up pulling up, what is this, stereolithography, 3D printer, pulling out of the resin. It looks like an organ, a liver model. Now, finger compression tests. He's squishing on it. Looks pretty, and then they perfuse vacuolar fluorescence staining of live cells. Alright, so this technology is actually 10 to 50 times faster than industry standard. And they're able to do pretty good resolution. And then they're injecting it with a vascular network. Now it's just a model. And how do you get stereolithography to print biomaterials? So they definitely have 3D printed human tissue and organs. Look at this. This is a 3D printed kiwi. No, it's like a kiwi that looks like a kidney. But there's a new device, and it's the extension of 3D printing. And are they talking about stereolithography again? No, doesn't really say too much, but pretty interesting to see the potential of stereolithography. If they can do what resins would allow for cellular, or maybe just accepting by the body. I mean, how do you get a body to not be rejected? Now, not sure why my live streaming isn't working, but let's see if I can pull this up and fix it on the fly, because I seem to be not live, and I'd like to be live. We do have a call-in number, just to remind you guys. Yeah, can't figure it out. But if you want to call in and leave us some happy news, some insight into what you'd like to say about the world, we do have a call-in number. It's 424-230-8808. Call in, give some thanks. Maybe we can give you a shout out live on air. And if not, we don't have to play it on live. Just say so. Nonetheless, thanks for listening and everything. We've got a couple more segments to get into, but just wanted to follow up. Okay, cool. So now we seem to be live. Whatever, I'm probably gonna upload this in a little bit, just the full rendering, but we've been talking about genetically engineered plants, GMO animals and regulations, talking about Aqua, Salmon, and Octopus prosthetics, 3D printing organs, biotech sensor, and how it's been growing. It's growing way too quick. And let's talk about tumors illuminated by and precisely with new biodegradable nanoprobes. Alright, so cancer are actually being labeled using bioharmoniphores. Now, this type of cancer, it's human, and they're doing it inside of a fish. Human cancer cells, 2 embryos, 3 embryos targeted, and then they put the 3 embryos inside and compared the two. And these photos are pretty impressive. Now, we do have a live chat, so if you want to join the live chat, feel free. I'm going to post it up again. Hopefully this will be good. Alright, so triphenylipidine and the peptide self-assembly encapsulates peptide assemblies. So you image single cancer cells by taking this peptide assembly and injecting it into the targeted biohormone. Now these targeted bio So, alright, lead researcher Dr. Perkis Panzanis of Imperial's Department of Bioengineering said, bioharmonics could be a more effective way to detect tumors than currently available. They uniquely combine features that could be great for cancer diagnosis and therapy in clinical practice and could eventually improve patient outcomes following future research. So bioharmonics are both biocapable and biodegradable as they are made of peptides. So they're metabolized naturally in the body within 48 hours and are therefore unlikely to pose long-term health risks. Alright, so these bioharmonivores, they don't bleach, unlike fluorescence. Now, if you're seeing what I'm pulling up, it's a artistic representation of the bioform structure. So it kind of has a molecular structure that is supported within the peptides, and they absorb light of a specific color like blue and then emit black light of a different color like green. However, tissues of the human body can emit light as well. Distinguishing the nanoprobe light from the background light can be tough and could lead to the wrong interpretation. So they developed these new nanoprobes and patented at Imperial, which emit light with a new type of glowing technology known as second harmonic generation. So after testing the nanoprobes in zebrafish embryos, researchers found that bioharmonics that were modified to target cancer cells highlighted tumors more brightly and for longer periods than fluorescent nanoprobes. Their light can be easily spotted and distinguished from the tissue generally emitted light. They can also attach precisely to tumor cells, no healthy cells, making them more precise in detecting tumor edges. Great article. Alright, so moving forward, let's move forward, right? Alright, so we talked about CRISPR and how does it move from the lab into our real life. And I think we're going to skip this subject. So we already beat the horse. But how tiny Indian team lets Uber launch medicines delivery in the US. Alright, so Uber Eats. You know it. They have actually a prescription-based company, Nimblerx. And it's been integrated into the Uber Eats platform. So COVID vaccines could actually be rolled out via Uber Eats. Let's just see, Nimblerx. Alright, so Nimblerx, they work with independent local specialty pharmacies to create delightful experiences for patients. So pharmacy delivered in as little as 1 hour. You can order online, quick delivery, save time. Click the link, place the order, get your prescriptions. Pretty cool. Secure payments. I like it. Alright, let's move on to this little
I didn't ask for this.
yeah, well, you're getting it. Here we go. This is a silicone-based laser. It's coming to be a reality soon. So science is actually inching towards using silicone more and more. And researchers from the Institute of Quantum Electronics now believe that this beautiful substance can be used to explore ways to integrate different functionalities in the form of semiconductors. Diode lasers, like the ones used in laser pointers and barcode scanners, are mostly based of gallium arsenide, which is abbreviated GaAs. However, GaAs, silicone, do not always work well together, but scientists have long awaited to realize the dream of laser on silicone. So, Professors Guillermo Salacari and Jerome Face from the Institute may have found a way. They have published in Applied Physics Letters in Focus Electroluminescence. In simple terms, is generating electrical light. Alright, so the silicone geranium, a semiconductor. Terahertz frequency band. I want photos. We have photos. No. I guess those are ads. Alright, so let's talk about this. Applied Physics Letters. This is a weird. I actually bring up. Why is this not a tweet? Boy. Wants me to tweet about it. Nope. Alright, so David Stark. Let's go to David Stark. Whatever. But it'd be pretty cool. Another reason to get into the silicon business. Institute of Quantum Electronics. Now, what is this? Well, I guess this is in Zurich. And it actually is big into Ethereum. ETH. Maybe not. Optical wiring for quantum computers. Well-formed disorder for light technologies. X-ray vision through water window. Sounds like a bunch of hot chaff. Photons, electrons, one-on-one. Alright, so I'm trying to figure out where this silicone. Okay, here you go. Lights on for silicone photonics. Alright. Terahertz, strontium device, step towards making silicone shine. So if you're looking at slightly two different frequencies due to the suboptimal injection of the upper state of radiative transition. Alright, that's great. Let's move forward. Alright, we got 3D printed organ technology may make transplant wingless a thing of the past. Yep, I should have read this earlier. But as for now, any person with failing organs is put on the wait list, ranked by the urgency of their situation. Some people never reach the top of the list and suffer the consequences by paying for their lives. If organs could be 3D printed within hours, the impact of the medicine world could be immense. Alright, let's click on this YouTube link. Why does it keep doing that? Oh, we already talked about this. Oh, wow, this is a full hand. They didn't even show the hand before. They just showed the University of Buffalo. Good job. Same article. Alright, scar-forming cells switch to the producing functional new neurons that promote functional recovery in mice after spinal cord injury. Now, I think this is actually really interesting because it could promote healing after a spinal cord injury. Functional recovery in mice after spinal cord injury. Scar-forming cells switch to producing new neurons. Wow. So they use genetically engineering techniques to trigger the production of functional nerve cells and scar-forming cells in mouse spinal cords in response to spinal cord injury. The study showed that the neurogenic reprogramming of the spinal NG2 glial cells led to functional improvements that the animals after SCI with newly formed neurons rebuilding neural circuits. Alright, Chung Li Zhang, Professor of Microbiology at W.W. Caruth, Junior Scholar in Biomedical Research, reported their results in a paper in Stem Cell, in which they can conclude our results reveal a cellular and molecular mechanism underlying neural injury-induced cell plasticity that can be exploited for adult neurogenesis and relay formation in a region that has largely lost its ability to regenerate. Let's see if there's any photos. Alright, so this graphical abstract shows a mouse with the spinal cord injury where the glia is actually affecting the neuroblasts, but astrocytes and ephemeral cells are not. So these neuroblasts are not translated to the neurons. Now, after the injection, the glia and the progenitors act on the neuroblast, which allows it to be attached to the neurons. Now, as the scar reduction occurs, there is a network restoration, allowing for the recovery of the mouse. Pretty cool. Pretty cool. Alright, one last biomedical engineering 3D. This is biomesh and the hernia repair complication. So hernia actually is one of the most common soft tissue injuries. Hernia is when formed when intra-abdominal content, such as a loop of the intestine, squeezes through the weak, defective, or injured areas of the abdominal wall. Alright, so this condition may develop serious complications. Therefore, hernia repair may be recommended. Repair consists of surgically implanting a prosthetic mesh to support and reinforce the damaged abdominal wall and facilitate the healing process. However, currently used mesh implants are associated with potentially adverse post-surgical complications. So although hernia mesh implants are mechanically strong and support abdominal issue, making the patient feel comfortably initially is common problem. After about 3 days after surgery, the implant can drive inflammation that into 2 or 3 weeks will affect organs nearby. So these mesh implants fail because they promote the adhesion of the intestine, liver, and other organs to the mesh. So as the adhesions grow, the mesh shrinks and hardens, potentially leading to chronic pain, bowel obstruction, bleeding, and poor quality of life. So some patients may require a second surgery to repair the unsuccessful first. So to address these complications, they developed a non-pharmacological approach by designing a novel mesh that, in addition to providing mechanical support to the injury site, also acts as inflammation modulating system. Alright, so this is opposites attract. This is a major innovation. So inflammatory mediators called cytokines appear when the mesh is implanted a few days after the surgery. Now, some of the cytokines in the implant, IL-1 beta and TNF, have positive surface charge due to the presence of amino acids lysine and arginine. So this biomesh with a negative surface charge would capture the positively charged cytokines, as opposed electrical charges are attracted to each other. We expected the trapping of the cytokines in the mesh would reduce their inflammatory effect and improve hernia repair and the healing process. So to test the idea, the researchers used a 3D bioprinter to fabricate biomesh of a polymer called phosphate cross-linked polymer, and through the experimentation, they optimized the mechanical properties so the mesh would withstand maximum abdominal pressure repeatedly without the deterioration of its mechanical strength for several months. They also showed that the biomesh did not degrade or reduce its elastic properties over time. It was not toxic to human cells. Cool. So as they degrade, they enable the mesh to capture more cytokines. Wow. Shin Labs. Alright, so last I want to talk about the blockchain. Alright, the biomedical blockchain research. Alright, what they do. So it's a Center for Biomedical Blockchain Research. It was established within Mount Sinai for the exploration and application of blockchain technology to problems in healthcare, biomedicine, and open science. So aims are be scholarly, make stuff, and translate understanding into action. Alright, so this domain expertise is biomedical, healthcare, emerging technology. Alright, so let's look at Joel Dudley. He is an investigator in translational bioinformatics and precision medicine. Joel is the Executive Vice President of Precision Health for the Mount Sinai Health System. And he's featured in Scientific American, CNBC, Wall Street Journal, Forbes, and other international media outlets. So resources, crypto tokens, contact, very general. I guess there's 6 of them. All right. So that sounds good. I guess we should know that blockchain, distributed ledger technologies for biomedical. So this is very general stuff, but it goes into maybe the problem of double spending without central intermediary. So it's about finance, invalid transaction, maybe the comparison of distributed network topologies. So to reject and yeah, I guess DeFi is very, very trendy right now. Anybody knows about DeFi? Let's pull up some DeFi stuff. Maybe later. Alright, so then you've got block caches, and then you've got node showing proof of works. Alright, so using Bitcoin as reference. Yeah, let's move on. Alright, so let's just get right into the BioHack. Alright, so if you guys didn't notice, we actually have a new Patreon. And I hate to harp on Patreon. I've never used it. Well, I actually support a couple people on Patreon, and I love it. I enjoy it a lot. Patreon's great. I do want to reinforce that, hey, I want to be more productive. And I want to make more videos. And there's 2 ways. I could release it on Patreon, or I could just start doing Patreon things on here. Whichever. We're not sure yet, but I want to get into specific biohacks, and this is my bread and butter. I want to talk about biohacks and what it means to be a biohacker. So, popular biohack is peptides, and it's a huge fitness supplement, anti-aging, and it can reverse the effects of aging, improve your physique. So it's actually a compound of amino acids, and so when a molecule of water is eliminated from the bond, it leads to a peptide bond, and they're just small proteins. So anything with 50 or fewer amino acids is considered a peptide. And yeah, dipeptides, 2 amino acids, tripeptides, 3 amino acids with 2 peptide bonds, and that's just how it goes. And our bodies actually make peptides from amino acids, so that's why it's important to get extra full BCAs, blockchain amino acids, just so you can have all the essential amino acids that your body needs to make to build muscle, fight against wrinkles, perform all these roles, because amino acids are the building blocks for protein. So your body needs to take in and produce amino acids in order for the peptides in their body to work efficiently. So I definitely supplement with the extra amino acids to make myself stronger, and to So glutamine and creatine are peptide supplements. So probably those have heard about creatine. Now, that is peptide. And they're saying, hey, why muscle size? Why you need to pair creatine with glutamine? So, get some gains, pair up the two biggest names. Alright, so creatine actually plays an important role in providing energy to muscle tissue stored within the tissues released during high energy demands, such as exercise. This is then converted to ATP, which is used as fuel to complete whatever energy demanding task you're doing. So it's also used for muscle recovery. So while glutamine is it's helpful for muscle recovery, post-workout inflammation, and a hormonal level to prevent catabolic breakdown of protein. So it's a muscle sparring supplement. Now, if you take them together, take about 20 grams creatine for 14 days, and then 5 days after that, and then 5 to 10 grams based on intensity, and then yeah, just swallow them whole. Now, peptides are pretty controversial, especially in my group, because you can actually inject them, and that would probably be the darker side of the world of peptides, which is like the promise of HGH. So human growth hormone. It's banned in popular athletics, and Peyton Manning allegedly took HGH to speed up his recovery from injury, and you get faster recovery, greater lean muscle mass growth, stronger injury recovery. So it's not some crazy synthetic substance. HGH is actually naturally produced, but certain peptides just encourage the body's production of HGH more than others. So by raising levels of HGH in your body, you can enjoy unfair athletic advantage over your competitors, which is why peptides that raise levels of HGH are banned. So you got 3 popular examples. IGF-1, GHRP6, Electroluminescence, and then there's injectable peptides like melatonin, which actually tans the skin. Let's just look at that real quick. Oh, look at this guy. Wow. Yep. He looks really dark. If you're watching on YouTube, this man is so dark from this peptide. I guess this guy's pretty dark too. Oh my gosh, look at that guy. Huh. I guess that's good. I think most bodybuilders will do tanning. And then instead, this is actually a way of looking. Let's just look at this real quick. He's just talking about whatever. Pretty interesting stuff. So if you want to learn more about peptides and maybe some of the anti-aging ones. Now, I've talked about before in my previous episodes peptides that could probably bring about clairvoyance. Now this is pretty interesting because how does this work? I don't know. And we're about to find out. So join the Patreon. I'm about to post a video of my experience, and hopefully we can bring up some more conclusions. Reduce the effects of aging in the skin. Make your skin look... Alright, effects of aging. So, Snap 8. I guess it's all injectables. Which is a very tough position because I'm sober, you know? I don't use pills anymore, so... Alright. There is a bottom line of peptides. Peptides are... you can get them from your BCAA supplements. You can get them from red meat, dairy, and other common ingredients. You can get them in anti-aging skin creams. So it's just a generic term. Nothing should be worried about. So, like I said in the beginning of the podcast, we want to analyze what this could do for you and what it means. You know, you don't have to be like this guy and sticking in your arm to get stronger, but there's a whole community out there, and we're gonna start digging into it. We're gonna do biohacks, and that's what the Patreon's for. We're gonna do tailored biohacks. We're gonna take requests. We're gonna do them. We're gonna build up a community. So there's nowhere we can't go. And I want to say thank you for watching. This has been a fun hour. Stay tuned. I'm going to try to do this every Friday at 4. Every Friday at 4 p.m. How's that sound? New show time. That's all, folks. Thank you. Have a wonderful night. And take care. Happy biohacking.